Welcome back to Biology Online with the Division of Education, Innovation and Energy e-learning program. I am your teacher, Lenetta Clarke, and today we are going to get right into part 2 of genetics. Last class, we looked at terminologies like allele, homozygous versus heterozygous, dominant versus recessive, genotypes and phenotypes, and we looked at simple genetic diagrams. At today's class, we would look at understanding pedigree charts, explaining the differences between codominance and incomplete dominance, and we will predict results of crosses from 1 and 2 above using Punnett squares. So we will start with pedigree charts. A pedigree chart shows the inheritance of genes or traits in a family over several generations. So here's an example of a typical pedigree chart. The horizontal lines will show or indicate a union or a marriage between two persons, and your vertical lines will indicate offsprings. So in this example, these two persons here had 1, 2, 3, 4, 5 children. This person here, this child here, got married to this person here, and they had 1, 2 children. Now most pedigree charts have a key. And in this particular example, the key here indicates who is male and who is female and what is the trait that we're looking at. So all our squares are our males, and all our circles are, of course, females. And for this particular example, we are seeing that the red shapes indicate the persons who have the trait or the disease that we're looking at, and the unaffected persons are the ones in blue. Now, pedigree charts can take several different shapes, and I'm just going to show you other examples of pedigree charts. So in this example, they would give numbers, and we would indicate, or we would assume, that the squares remain the males and the circles remain the females. In this particular example as well, they show the generations on the side. So here's generation 1, 2, 3, and 4. So for example, in generation 2, we have 1, 2, 3, 4, 5, 6 individuals. Individual 1, 3, and 5 are offsprings of the first union from generation 1. So this is another example of a pedigree chart, but in this case, each person has their own individual number. And we'll use this one to go straight into our first activity, dealing with pedigree charts. So let's look at this question. Cystic fibrosis is an inherited condition. The diagram shows the incidence of cystic fibrosis in a family over 4 generations. Number 1. What evidence in the pedigree suggests that cystic fibrosis is determined by a recessive allele? Now, one answer that we can give, if you look at the diagram, we are seeing that person 4, person 9, 16, and 17 all have the disease of cystic fibrosis. However, most of the individuals in this family tree or this pedigree chart do not have the disease. So one response could be just by the sheer numbers of persons who don't have the disease, we can assume that cystic fibrosis is not a dominant allele trait, but it is a recessive one. Also, we know that because an individual receives alleles from both parents, in order for you to have the disease, you must have gotten a recessive allele from both parents. And therefore, when an individual has a disease, they would have gotten both recessive alleles, and therefore they will have the trait for the disease. Question 2. What is the genotype of individuals 5, 6, and 9? So we are going to use the letter F, the dominant capital F, to represent normal, and we will use common F to represent having cystic fibrosis, as in the disease. So the question asks, what is the genotype of individuals 5, 6, and 9? Individual 5 here does not have the disease. However, that person has a child that has the disease. So the genotype of individual 5, if an offspring of individual 5 has the disease, it means that that person, or this female, it's a woman, this female would have passed on one recessive allele to her offspring. So this person, this female here, her genotype must be capital F, common F. She is capital F, common F because the dominant allele, capital F, suppresses the effect of the common F. 5, 6, individual 6 is also similar to individual 5 because individual 6 also has a child that has the disease. So both, it means that both parents would have donated a common F or a recessive allele to each of their children. So individual 6 genotype will be capital F, common F. Let us look at individual 9. Individual 9 is easy. Individual 9 has the disease. So therefore it means that individual 9 does not have any dominant alleles in their genotype. So individual 9's genotype is common F, common F. So that person has cystic fibrosis. Number 3, draw genetic diagrams to work out the probability that the next child born to individuals 8 and 9 and 11 and 12 will suffer from cystic fibrosis. I would like you all to take a minute to work out your answer, or you may pause the video now or take a screenshot and work on your answer during the break. Number 3, draw genetic diagrams to work out the probability that the next child born to individuals 8 and 9 and 11 and 12 will suffer from cystic fibrosis. So we're looking at individuals 8 and 9. So let's just go back to the pedigree chart, and we're seeing individuals 8 and 9 here. Remember, we're using the letter F, capital F for dominant, and common F for the recessive trait. So individual 9, we had already worked out that individual 9 has a genotype of common F, common F. And if we look at individual 8, we are seeing that this person does not have cystic fibrosis. But we don't know if individual 8 is homozygous dominant or heterozygous for the trait. So in order to figure that out, we also need to look at the offspring. And we are going to have to make a big assumption here because none of the offspring have cystic fibrosis. So we're going to make the assumption in this case that individual 8 does not have any recessive alleles to pass on to the next generation. So let us use, going back, capital F and common F to represent the alleles for the trait cystic fibrosis. So the parents' phenotype, we know one is normal, and we know that one has cystic fibrosis. Parents' genotype. The parent that has cystic fibrosis is a male, cystic fibrosis. So we use common F, and we are using the assumption that the normal parent does not have any recessive alleles to pass on to the offsprings. So the gametes in this case we know are all capital Fs for this individual and all common Fs for the other individual. Random fertilization, and the F1 genotype will be capital F, common F. Or in other words, they would all be normal. So the question asks, what is the probability that the next child born will suffer from cystic fibrosis? And in this case, the answer is 0 or 0%. There's no chance that the next child born to individuals 8 and 9 will have cystic fibrosis. Part 2 asks, what is the probability that the next child born to individuals 11 and 12 will suffer from cystic fibrosis? So let's look back at our pedigree chart. Individuals 11 and 12 are here, and their phenotype indicates that they are normal. They do not have the disease. However, we are seeing that from that union, they have 2 male children who have the disease. It therefore means that each parent has a recessive allele that they passed on to their children. So, hence, both 11 and 12 are heterozygous. So we have 2 normal parents not suffering from the disease, but their genotype indicates that they do have a recessive allele each. So they're heterozygous, and therefore the gametes that they would have would be, some will be capital F, some will be common F. Some of the gametes here will contain capital Fs, and some gametes will contain common F alleles. And as usual, we look at all the possible combinations that these gametes, when they meet and fertilize, would create. This was covered in part 1. So we'll go through this quickly. Now, if this is confusing to you, be sure to look at part 1 of our previous version of genetics. So the genotypes of the F1 generation here is listed on the board. So we have capital F, common F by 2, and homozygous recessive, meaning common F. So the phenotypes of these children will be, all these here will be normal because of the presence of that dominant allele, and this one will have cystic fibrosis. So let's look at the question. What is the probability that the next child born to 11 and 12 will suffer from cystic fibrosis? So there's a 25% chance that the next child born to the parents of this union will have cystic fibrosis. So it's a 1 in 4 chance of having cystic fibrosis. So let's continue. Incomplete dominance. Now, in previous examples, we have been working with complete dominance, where the dominant allele completely suppresses the expression of the recessive allele. However, there are cases when that is not always the case. And we're going to look at cases of incomplete dominance. So for this example, we have a white flower crossing with a red flower, and the result is, shockingly, a pink plant. Now, we know that in the past, it would either be red or white. But sometimes we do have these anomalies occurring in nature. And this is what we call incomplete dominance. Incomplete dominance, the dominant allele does not completely suppress the effect of the recessive trait. The result is a blending of both traits or both phenotypes in the heterozygous state. So that is why we have a blending. So we have red and white, we know when blended together makes pink. So in incomplete dominance, the dominant allele does not completely suppress. So we have the two colors will merge together to create a color of pink. So quick quiz. Which is not an example of incomplete dominance? A pink flower produced from red and white flowers. B, a flower that is both red and white produced from a red and white flower. C, curly-haired and straight-haired individuals producing a wavy-haired offspring. Or D, a highly spotted dog and a non-spotted dog producing puppies with a few spots. I give you 2 seconds. The answer there is B. This is the only one that is not an example of incomplete dominance. As we said before, when you have incomplete dominance, you have a mixing of colors. So we have red and white flowers creating a pink flower for option A. In option B, we have curly-haired and straight-haired individual creating a wavy-haired offspring. So that means that there's a blending of both traits. And in D, we have a highly spotted dog with a non-spotted dog creating a dog with few spots. So that's another example of blending of the two traits. So let's work on an actual CSEC type question on incomplete dominance. A rose shows incomplete dominance. So in this question, they are telling you that it is an incomplete dominance question. When a red flowered plant is bred with a white flowered plant, the offspring are pink with the phenotype capital R, common R. If these pink individuals are crossbred, what will be the ratio of their offspring? So you can take a screenshot at this point, or you can pause the video and come back in 1 minute and we'll discuss the answer. Okay, to answer this question, I'm going to introduce the concept of Punnett squares one time. So we are going to use the letter R to represent the flower color, and we have capital R for dominant trait and common R for the recessive trait. So let me just remind you of what the question was. We have a red flowered plant being bred with a white flowered plant, and all the offspring in this case were pink. And they have now asked us to cross these pink individuals and see what the ratios of their offspring will be. So the red flower plant crossing with a white flower plant would produce genotypes of capital R for red flower and common R for white flower. The gametes, of course, are all capital R for this plant and all common R's for this plant. And in a Punnett square, it's quite simple. You literally draw a square and you put the gametes for one parent at the top and the gametes for the other parent on the left. So we have capital R, so we put these gametes here, and the common R gametes for the other parent are on this side of the board. And let us look at what the results for the offsprings will be. So everything in this box would be a result of this capital R crossing with this common R. For this box, we are looking at this capital R with this common R. Again, we are looking at this box here, capital R and common R. Similarly, we're looking at this box here, capital R crossing with common R. Now remember, this is a case of incomplete dominance. So we have the dominant allele does not completely suppress the recessive allele. So we are going to have all pink flowers because we have blending of both the red and white traits. So all the individuals from the cross of red and white flowers will have pink genotypes, as you can see here. Now, we have not actually answered the question as yet. The question asks, we were already told that we would have pink individuals. What we want to know is if we cross these pink individuals, in other words, if we make them self-fertilize, what will be the ratios of that offspring or those offspring? So that will be your second generation. So let's look at the Punnett Square showing the cross between pink flowers and pink flowers. So once again, another Punnett Square. We have the genotypes of capital R, common R for one parent and capital R, common R for the other parent. And we're looking at the result of the offsprings here. So in this box, we have capital R, common R to give you, capital R to give you homozygous dominant. For this one, we have capital R and common R. So we have a heterozygous case, common R, capital R, another heterozygous case here, and common R to give you a homozygous recessive case. And what will be the phenotypes? Capital R is red, common R is white, but in the heterozygous state, where you have capital R, common R, the flowers are pink because the dominant allele of red does not completely suppress the recessive allele of white. So we have blending of red and white, so we have pink flowers. So the ratio of the F2 generation is 1 red to 2 pinks to 1 white. You are free to pause the video and recap to ensure that you understand before moving forward. Now we're going to look at codominance. Codominance is a little different from incomplete dominance. So if you look at the image on the screen, we have a red flower and a white flower producing in the left corner at the bottom an incomplete dominance case where we have pink flowers, and we just explained that one. However, sometimes you will see red and white flowers offspring from a red and white crossing. And from the picture you can see that in the codominance case you have the existence of both colors showing up in the offspring. So not a blending, but it's a sharing of the two colors on the offspring at the same time. So think about like co-owners of a business. Both business owners have 50% share in the business. So in this case, you have both colors will show up in the generations after. So in codominance, both alleles of a pair are dominant and equally expressed in the heterozygous state, which is the phenotype. And a perfect example is blood groups in humans. In humans, we have the ABO blood system where 3 alleles code for blood type. Both allele A and allele B are equally dominant. However, allele O is the recessive allele. So let's look at blood type and how we go about determining whether you're blood type A or blood type B. So I mentioned before that we have 3 major alleles in our blood system, blood typing system. We have allele A, B, and O. However, A and B are both equally dominant. So for someone who has a phenotype of blood A, so what you will see or what you will say is that you have a blood type of A. However, your genotype, what is on your genes, you can either be, let's say that is where your blood type gene is found. You can either be capital A, or you can be capital A, O. Because A is dominant to O, so what is expressed would be, your genotype will be AO, and your phenotype will be blood group A. If you're blood group B, similarly, you can either be capital B, homozygous dominant, or capital B, O. B being dominant to O, so what is expressed would be blood type O. If you're blood type A and B, because A and B are both dominant, they would both show up in the genotype. None is dominant, completely dominant over the other. So you'd have A and B alleles at the same time. And finally, if you're blood type O, because this is the recessive allele, the only option to express phenotype O will be O. Just another example of codominance in animals. We have red cattle, which is denoted by the capital R, which is homozygous, and we have white cattle donated by the genotype W. And so in the heterozygous case, because they are both dominant alleles, you have a new color of cow that is called roan, and it is a red and white cow in terms of its phenotype. So this is another example of codominance in cattle. So let's look at a question. Aaliyah's blood type is A. She has a son whose blood type is AB. She has named Kes the father of her child. Kes's blood type is O. Use the Punnett Square to prove or disprove Aaliyah's claim. Please pause the video now or take a screenshot and attempt the question on your own, and we'll come back in 1 minute to see if your answer is correct. Is Kes the father of Aaliyah's child, or is he not? Okay, so what did you get? What did you arrive at? Is Kes really the father, or is he not? Let's see. So Aaliyah's blood type is A. So let's just write Aaliyah is A, and the son is AB, and Kes is blood type O. Blood type O. So let's see. Because Kes is blood type O, then we know that his genotype can only be O, because it's the recessive allele. So we'll put Kes's alleles here, or his gametes rather, O. Now Aaliyah, she has blood type A. Now the only way she can be blood type A, or there are two options for her. Her genotype can either be AA or AO. Those are the only possibilities that she can have to have a genotype or a phenotype of blood group A. So let us look at both of them and see if Kes is really the father of her child. So we will use this one first, and we'll put the alleles here, capital A, capital A. And what are the probabilities of all the offspring that she and Kes can have if her genotype is capital A, capital A? So we have AO here, AO there, AO there, and AO there. Now the son's genotype is AB, and his phenotype as well. So in this example, if she's genotype capital A, then there's no way that Kes can be the father of her child. So let us look at if she were AO. If her gametes some of her gametes had A alleles and some of her gametes had O alleles, let us look at what would happen. So we'd have AO here, and over here we'd have OO. So it looks like Kes is not the father of Alia's child. So that brings us to the end of today's class. We looked at pedigree charts, we introduced the concept of Punnett squares, and we used Punnett squares to determine or predict the outcome of offsprings between incomplete and complete dominance and codominance. So I urge you to please go back, and at the end of this program, you will have a list of activities to attempt on your own, and please ask your tutor or your local teachers for assistance if you are stuck. Thank you, and please join us for part 3 of genetics.